First of all, my thanks to Dr. Rao and Dr. Ramkrishna for inviting me here. One thing all of you would have noticed with my previous speakers is the passion with which they spoke. The passion about the technology, the passion about the science that they have. And what I intend to do is show you that it's really important to have that passion. Money will come. Money will automatically come for you to do the work. So I'm going to talk to you about something which is of high socio-economic impact in India. This is going to be extremely challenging. I'm going to talk about neglected tropical diseases and the work that we do at my institute in Bangalore, which is called the Foundation for Neglected Disease Research. So started this institute in 2014 when the AstraZeneca Research Center in Bangalore closed down. I used to be the head of R&D there, and financial support and in-kind support from them, I started this facility, which is a one-of-a-kind, not-for-profit organization, which works on developing new drugs for diseases which have a high impact in India. We work on mostly on the infectious diseases. We probably are the only biosafety 3 lab in the country, in the private sector, which has the capability to do animal research. Though my friend Manny would tell me, reduce the number of animals that I use, but there is a place for animal research in drug discovery. So we work on a variety of areas, mainly bacterial, fungal, viral infections. This is the pipeline that we have of drugs. So, if anybody tells you drug discovery and development in India is not possible, send them to me. We beg to differ. We have discovered drugs in India. We have discovered drugs which have become drugs globally across the US, Europe, and in India. So I don't think that it's difficult to do drug discovery in India. It just requires perseverance, patience, and a lot of good contacts to get the right things done. Yeah. So this is the pipeline of drugs that we have, and I especially want to touch about two things. One is TBA 7371, the one on the far left corner there, which is a drug which was discovered in India, is a novel mechanism, a first-in-class opportunity, which is being tested in TB patients in a phase 2 clinical trial. We completed the phase 2 clinical trial in South Africa, and now we're trying to bring this back to India for a phase 2B clinical trial. Chloroquine, you're all aware of, and a variety of other drugs, including newer technologies like BAC Protax for infection, which not many people in the world have embarked on. So what I want to tell you with this slide is that it's possible to do drug discovery. It's possible to work in areas which are not being worked on globally, and we can make a difference. So this is my small R&D center in Bangalore, which is about 10,000 square feet of R&D space, including the BSL2, BSL3. We do the entire length and breadth of drug discovery and development, from our own proprietary chemistry all the way up to animal efficacy. We don't do any of the GLP tox studies, but everything else is done in-house. And we have some of the state-of-the-art equipment that is available anywhere in the world. So that's me and my co-founder. Both of us were at AstraZeneca. We have a team of about 25 scientists who deliver all of those projects. So we work extensively in collaboration. We work extensively in collaboration with Indian academia, with Western academia, with industry, you name them, and we work with them. So I want to talk about neglected tropical diseases today. And this is a topic which is very close to my heart, and one where we really want to make a difference. What are neglected diseases? Neglected diseases are a diverse set of bacterial, viral, fungal diseases which are having a high social impact and a significant impact on the health. They are primarily affecting the poor, the marginalized, and the vulnerable populations, and especially where the social hygiene is limited. Why are they called neglected? They are neglected because most pharma companies would not work on this, because they have a very low commercial value. They have high social impact, low commercial value. And the challenge is most of these diseases are often misdiagnosed. So the right treatment is not given to the people, right? And this is what we want to change within our organization. Now, in many forums that I go to, I keep getting asked the question, are you working on rare diseases? There is a difference between a neglected disease and a rare disease. And I want to bring that out very clearly, right? Neglected diseases are those which receive limited attention. Almost 1 billion people on the face of the earth suffer from at least one neglected disease. So that's the extent of the neglected disease. There are 21 such diseases which are recognized by the WHO and they are endemic to developing countries. On the other hand, rare diseases are those which impact less than 1 in 2,000 people in Europe, fewer than 200,000 in US, and about 1 in 5,000 people in India. There are about 7,000 documented rare disease cases across the world, and they are prevalent. And the case is that if you have a drug for a rare disease, you can sell it for millions of dollars, right? The cost of therapy, a single therapy can be $1 million. So that's the difference between what a neglected disease and a rare disease is. Most companies would want to work on a rare disease. We chose to work on the neglected disease. So this is a list of the WHO neglected tropical diseases. You have the bacterial, viral, and the fungal, and a lot of the parasitic diseases as well. One clear thing that has come out in the recent past is the snake bite as a neglected disease. If you look at the heat map of the world, you will see that these diseases are highly prevalent in the southern part of the world, right? Africa, Southeast Asia, South America, these are the hotspots for many of these neglected diseases. There are at least 184 countries in the world which have at least one neglected disease as an endemic. In India, we have 5 of them, at least, recorded. We account for almost 40% of the total burden of neglected tropical diseases taken together. And highly prevalent in India, leprosy, trachoma, dengue, chikungunya, rabies, and a variety of others. And today what I want to talk to you about, rabies, right? And most of us are aware of this. Show of hands, how many of you are aware of rabies? Yeah, almost everybody, right? How many of you think there is a drug available in the market for rabies? Is there a drug available which can treat rabies? The answer is no. Prophylactic vaccines, you have monoclonal antibodies which are given, but there is not a single drug which is today approved for the treatment of rabies. And this is what we want to change. So this is a program that we've just started within my organization. I'm going to share some of the work that we do and hopefully would entice some interest from you guys, the students in the audience, to come and want to work with us. So it's basically caused by a Lissa virus called the rabies Lissa virus, and is a single-stranded RNA virus. It is one of the viruses which is highly neurotropic. It has a high affinity for the nervous system, and that is why it is so deadly. The fatality rate is almost 100% as compared to any of the other viral diseases. This is one where if it reaches the brain, it is almost 100% fatal, right? It is also 100% preventable by a vaccine, right? So if you take a vaccine in time, you could actually prevent the disease. But I will show you why it is still important for us to work on something like this. And the first case of rabies was treated by a vaccine by Louis Pasteur and his colleagues, and the child was called Joseph Meister, who was bitten by a stray dog. So the rabies virus, according to its fatality ratio, is actually shaped like a bullet. So it's a single-stranded RNA. You can see the genome helical coil, the outer matrix protein, and the right side shows you how the virus actually gets in. So you have a first stage of where it binds to the receptors, which are the nicotinic acid. I'll come to that in a minute. Shows the penetration, gets in, the viral coat is removed, and then there is a division of the RNA, and then the budding happens. So this is the basic structure and replication of the rabies virus. This is how it gets into the brain. So you have a bite from a rabid animal. The virus is inoculated into the muscle. In the skeletal muscle, it is replicating, travels within the axons to the peripheral nerves, and gets from there into the brain, from the motor neurons. So basically, the nicotinic acetylcholine receptors at the neuromuscular junctions are the key ones which are helping the rabies virus get into the system. The consequence of a rabies virus infection include extensive damage to the central nervous system, which results in coma and death. It's prevalent in more than 150 countries across the world, and 95% of these are in the Asian and African countries. Almost 60,000 deaths a year, of which 35% occur here in India, right? So there are about 20 to 25,000 deaths from rabies even today in India. And that's the population that we want to target, right? We want to prevent those 20 to 25,000 deaths. It's often misdiagnosed or underdiagnosed and hence underreported. And in India, primarily dogs are the main reservoir of rabies. So this is a heat map of how the dog-mediated rabies is across the world, and you can see again the southern hemisphere is one where there is a high prevalence of something like this. There is a very dramatic clinical presentation with rabies, right? So you can see a patient who's been affected and the virus has reached the CNS system. At the sight of water, even just fanning with a newspaper, it tends to create a fear response. You have pharyngeal spasms, and you also see a lot of aggression with these patients. And the last final thing is hydrophobia. And this is where even at the sight of water, there is a significant fear response that is elicited from the patient. There are basically two kinds of rabies, encephalitic rabies and a paralytic rabies. So here is one where the patient goes through fluctuating consciousness, a lot of spasms, which are signs of autonomic dysfunction. You have hydrophobia, which is one of the most characteristic features of encephalitic rabies. That is when the CNS is affected. You have a paralytic rabies where the limbs are paralyzed over a period of time, and especially even including the respiratory muscles. There is significant piloerection and edema that is seen. Hydrophobia is uncommon in cases of paralytic rabies, but it may actually be seen in the terminal stages where there is a involvement of the CNS as well. So this is the duration of the rabies over the period. So you have a 20 to 90 day incubation of the virus depending on where the bite is and how long it takes for the virus to actually travel to the central nervous system. But once it reaches the central nervous system, the resulting is less than 10 days, right? So once it has reached the CNS, it multiplies very fast. You have an acute neurological phase that is about 1 to 4 days, which has several of these physiological changes that you can see, resulting in coma and finally death within 7 days. The challenge with rabies is it doesn't get detected very early, right? Just because of this long duration. So once the dog has bitten, people wash it away and they say, okay, no symptoms, 2 days, 3 days, there is no symptom. They don't take the vaccine. And what happens is when they go to the clinic, they are also presenting many other symptoms which actually lead to a misdiagnosis. So you have things like encephalopathy, a variety of metabolic disturbances, drug addiction or illicit drug use, variety of other bacterial and viral infections which resemble some of these symptoms. Now, as a result, the diagnosis is much lower. So you have two types of diagnosis which are done, the antemortem and the postmortem. For the antemortem, you have three different techniques using viral RNA, the DFA, and also a serology. Whereas with postmortem, you can do even the brain histopathology to look for Negri bodies. So you can see very clear, this is a postmortem slice of the brain. You can see very clear Negri bodies that are identified within the various regions of the brain. So if you have a suspected or a confirmed case of human rabies, there's either a palliative care at home with lot of sedation, rehydration, analgesics, all of that, or an aggressive management in the hospital that is done depending on what that case looks like. So in terms of current treatment, prevention seems to be the best defense, and there is a pre-exposure prophylaxis and a post-exposure prophylaxis that can be done. Contains giving multiple doses of the vaccine, right? Wound care with soapy water, scrubbing to remove the foreign bodies, use of iodine-based antiseptics, and a post-exposure prophylaxis with an immunoglobulin and a vaccine. So this is the current treatment that is available. So you have either the immunoglobulin from the horse or the human, and you have the rabies shield, which is a monoclonal antibody, and a twin rab from Zydus, which is two monoclonal antibodies, a combination, and vaccine, which is the Vaxirab, also from Zydus. So this is the entire picture of what is available today, and there is no small molecule drug that is on this list, right? And today, what we want to see is if this is not available, then what is the solution? So I just skip through this for interest of time. Right, come to this. So what are the challenges? So one is the vaccine cost, which limits its accessibility and availability. The efficacy of the vaccine being affected by the cold chain. So this needs to be maintained at 2 to 8 degrees. So if there is no cold chain, the vaccine deteriorates or the efficacy is reduced. And no drug available today but this is an opportunity more than a challenge. So the solution that we propose is to discover highly potent small molecule antivirals that can be used either prophylactically or therapeutically. So what are the challenges? It's a neglected disease, so there is no funding, right? I've put the word low funding to be politically correct, but in reality, there is no funding. It is endemic to LMICs, so the market size is limited and there is a low return on investment. So the commercial value is definitely low. There's a challenge to the drug design. If you want to treat rabies, you have to get into the brain. And to get into the brain, designing a drug is not that simple. So the CNS the penetration of the blood-brain barrier, even in a case when it is inflamed because of rabies, is still a challenge. Getting to an assay which can actually screen the compounds is even more difficult. Since the accidental exposure cannot be detected very easily, every single person who works in a rabies lab has to be vaccinated. And the vaccine antibody titer has to be measured every 6 months to make sure that they have sufficient antibody titer to prevent any accidental infection. We have a small group of 5 people who work in the rabies lab. Only they are allowed to enter. Nobody else enters that lab, and all 5 of them are monitored continuously for the presence of antibodies, that in case there is an accidental exposure, though we take all the precautions required, still that they may not be affected with the disease. The clinical development is very challenging because nobody has done this before. So this is a territory where no one has gone before, right? Like the Star Trek serial. So we definitely want to go down that path. So there are a variety of drugs that have been tried. You have seen, including some of the antivirals. There is one Milwaukee protocol that was followed, which showed some effect, but again, has not been efficacious in variety of populations. Today, there are about less than 35 documented survivors worldwide who have survived after the rabies virus has reached the brain. In India, there are about 20 of them. Most of them had had a previous rabies vaccination or a robust antibody response, and they have tested negative for the viral antigen. Based on this, we believe that there is a potential for a new therapy to come in. There is also a chance, which we are seeing more and more, that in spite of the rabies vaccine being taken and the course being completed, patients are dying, right? So there has been a recent case in 2024, which was recently, about 10 days ago, here in Visakhapatnam. There has been a death after a pet dog has bitten the father and son, right? 2 people have died of rabies. So there is a significant increase, if you look at the numbers, right, of dog menace in the country and the bites per day. There is a significant increase in the number of bites per day as we have moved over the last few years. And this is a cause of concern because this can lead to a significant increase in the number of rabies cases. Touched upon this earlier. So what should a new rabies drug look like, right? So this is our ideal target product profile that we want for a rabies drug. Obviously a novel and patentable drug with a variety of parameters which have drug-like properties. So most of you, if you're pharmacy students, you know what a drug-like property is, according to that. We have 2 strategies. We work with repurposed drugs and we also work with new chemical entities. We have an FDA-approved compound library. We're looking for drugs which are CNS penetrating, so that library number is small, but we do a complete screening using our in vitro screens. And we also have our own compound library, which is a proprietary compound library, which we are looking at screening in this assay. Till date, we have screened about 3,000 compounds and we've got about 29 hits. And this is from the repurposed library. And you can see the distribution of where the hits are coming from, right? So you have a majority of them coming from the cancer, a lot of them coming from the infectious disease space, and a variety, 14% of CNS drugs which seem to be active. So out of the 29, we have at least 2 or 3 of them which have the ability to cross the blood-brain barrier. So we are very hopeful that these could either be good drugs themselves or could provide very good starting points for us to do some novel chemistry. So just an indication of the assay that we have done. We have looked at drugs like clofazimine, niclosamide. These are a couple of the hits that we have. So what we want to do is use our institute to actually renew the interest in neglected drug disease drug discovery, and to use drug repurposing as a premier strategy for us to do the target is to have 0 deaths by 2030 from rabies. This is the WHO goal, right? And this can happen through mass dog vaccination, improves PEP, and bite case management. But I think there is a significant role for small molecules to help in achieving this goal. Thank you very much for your attention. I'll leave you with this last slide.